Hi, everyone. I'm Faye Minty, mom to an 8P hero. And this is another Project 8P quarterly update. This time we're joined by Dr. Joe Bellucci. Have I got that right?
That's right.
Okay, great. Who earned his PhD in biomedical engineering at Duke University. He is the director of high throughput biology at Rarebase and is helping to identify drugs which may help our 8P heroes. Now, to put Joe's work in context, I'm going to give a tiny recap of the family-friendly research talks. Now, the Columbia University study, Wendy Chung's study, established that the commonly deleted interval among 49 of the 8P heroes was the 6.7 megabase end of the 8P chromosome arm. Now, that's shown in the cartoon here on the right-hand side, one normal chromosome 8 in green, the chromosome with the genetic anomaly in orange, and the deleted section in red. So that section in red is gone. That's no longer present in these 8P hero cells. Now, CSMD1, the region I'm showing there, is a massive gene, one of the biggest in the human genome. And this is just one of 14 genes of interest that have been identified in this deleted region. One of the modes of gene expression is the process of genes becoming proteins. In the examples shown previously, the deletion would mean that we wouldn't have enough protein, so not enough gene coding for protein. And I'm showing that here as only having 3 green triangle proteins instead of 6. So maybe some human biological processes will continue as normal as a consequence of this deletion, but some might not. This is part of the unknown of our 8P heroes. So just imagine if we could give our 8P heroes a medicine that could correct that loss, something that is capable of increasing the amount of protein from the critical genes, from the remaining normal chromosome 8P arm, and this helped to improve some of the symptoms we see in our 8P heroes. Now, a key safety factor in this process is making sure that if a drug is discovered that can do this, that it doesn't upset the balance of all the other proteins which are required for human function. Now, during the Project 8P roundtable in March, Joe showed this image on the left-hand side. So here, Rarebase have grown some cells in the lab and added a drug to see how this affects the gene expression. So what we're looking at is just one possible drug of many screened to see what happens in this instance with CSMD1 gene expression compared to the expression of 12 other genes. So Joe, can you tell us, do I have this right in the schematic on the right-hand side? This would be, sorry, I've lost my place here. This would be the ideal pattern in each of those plots on the left. Lots of CSMD1 gene expression on the right-hand side, where I have these purple smiley faces, and nothing in the box at the top, which would represent an increase in a different gene being expressed.
That's exactly what we're looking for here. So these 12 genes that are shown on the left are what are called housekeeping genes, and they're genes that should have a consistent level of expression across different cell types. So we wouldn't really expect these to be moved either up or down by any of the compounds that we're testing. You do see that for many of them. But what you're describing is right. In an ideal scenario, best case, we would try to identify a compound that is moving the gene of interest specifically. In this case, CSMD1 is what you're showing on the right. That's actually not the case for most small molecule drugs. We and others have shown that, most FDA approved compounds do move a large number of genes, both up and down. In our data set, it's around 600 genes on average that a compound will affect. But I think the important thing to keep in mind is that selectivity, that CSMD1 alone going up in this case, it's kind of like the ideal scenario, but it's not necessary in all cases. These FDA approved drugs are, many of them are safe and effective. They're still moving this large number of genes, but our hypothesis is that the sort of movement of the disorder gene is kind of most important and has the most biological effect, where those other genes are actually not really processed by the cell in exactly the same way. They're not necessarily having a bad function on the cell. So really just the recovery of the disorder gene is kind of the main thing we're looking for at this point.
Mm-hmm. And it could be that there's other genes which wind up, being affected and also having their proteins expressed more, upregulated, but it could be inconsequential, right?
That's right. And we obviously have tools to sort of identify things that are clearly not good to move. Like we would not want to move genes that are known drivers of oncogenesis, for example, things like that. We can also do that at the kind of biological pathway level, right? We wouldn't want to make the cells more inflamed, for example, and we can actually measure that in these types of data sets.
Perfect. Thank you. So how many drugs have you screened so far that might possibly drive an increase in CSMD1 gene expression?
So I think what's shown here on the left is that we do have a set that we'd like to follow up on. I think it's important to keep in mind that this is just sort of casting a wide net in this screen. We've only tested each of these compounds one time, so there will likely be things that don't replicate in follow-up studies. It's just kind of the nature of the business here.
Yeah.
But there is a set of around 10 that we'd like to move into that follow-up testing to determine whether they're actually having an effect or not.
Really good. Great. So that's perfect. That takes us on to our next slide. So in this example, with the goal being identify and evaluate candidate compounds that upregulate CSMD1 gene expression, these are the experimental questions that you mentioned you would move on to. So milestone 1, concentration. And that's really what dosage of drug you would use in experiments. And correct me if I'm getting it wrong here, Joe. And milestone 2, function. So this is not just showing that there's more protein, but that, again, it has a consequence, right? That it's interacting. If you think back to for the families, if you think back to the triangles, that if you added those three extra triangles, it would actually cause something. It would react with that chevron to move the process forward, and they wouldn't just sit there looking at each other going, whatever,
Yep, that's exactly right. In milestone 1, we're basically looking to see whether any of the compounds have a drug-like behavior. And what that means is that you should see, as you increase the concentration of the compound, kind of an increase in effect on gene expression of CSMD1 in this case. Milestone 2 is really important because that kind of connects that change in gene expression to actual functional improvement in the cells. And this is a little bit more difficult to tease apart and is really disorder specific. But I kind of equate this to like an analogy with a car. If you've got like a flat tire, you're trying to put more air into that tire to pump it up. It's kind of the same thing as what we're doing with protein. You should be able to measure the functional effect of putting air in the tire by looking at fuel economy, for example. Fuel economy should go up if the tire is at the right pressure. So that's kind of like the idea of what we're trying to do here, is not just look at what the pressure of the tire is, but also does it affect the overall performance of the cell in this case.
Yeah, perfect. That makes a lot of sense. So let's just see so with each of these drugs, what you're talking about here though is trialing the drugs in the 8p hero cells in the lab, right?
Yep,
so
that's right.
Not straight to 8p hero.
Correct. Yeah, we actually do sort of our baseline neurons first. These are sort of non-disorder affected, presumably healthy cells. We do the candidate confirmation in those, and then we go to the 8p hero cells in the lab, and then that will motivate further evaluation of any of the leads that come out of that process.
Perfect. And so I just want to emphasize that in these slides, CSMD1 has actually just been used as an example. It's the most commonly it's the most common example that we go to. But there are other candidate genes, so that's DLGAP2 and ANGPT2. which are also in that deleted region. And with regard to the extra gene expression we see in AP heroes who have duplication or inverted duplication deletion, can you tell us, Joe, will you also kind of move on to look for drugs that can downregulate their gene expression? I think you mentioned it earlier, yeah? And that would be the opposite of like essentially the way you've been working here, is that right?
Correct. We measure essentially all genes in these cells as they're being affected by all of these different compounds. So we have the opportunity to look at a variety of genes, not just CSMD1. The RNA-seq data that we have obtained does suggest that there might be a couple of other genes that are regulated differently in the AP patient cells versus the matched control. So there are other routes that we'd like to explore, not just CSMD1.
Perfect, thank you. And that really just, I'm just going to mention real quickly to the families that if anyone wasn't quite sure what I was talking about with the background of understanding the study, if you go to the YouTube channel, you'll see there's the family-friendly presentations. You can just click on those and watch through, and anyone can get in touch with me through Project 8P. I'd be happy to explain more if it would help. And really just to say thank you to Joe. Thank you, I mean, mostly for all your work, for joining us for today. And thank you to all the families for watching, for continuing to complete their surveys in the Project 8P registry. And if there's any families out there feeling confused, not sure how to proceed with the registry, get in touch with Katie. She's had to help me quite a lot in the registry, and she's an expert in it now. So thanks, everyone. Thank you, Joe.
Thank you.
Yeah, cheers.